Hello students, in today's class we are going to learn about different types of blotting techniques. So blotting techniques are basically techniques that are used for transferring DNA, RNA or protein molecules onto a membrane where it can be used for detection of specific DNA, RNA or protein molecules. So the southern blotting technique is used to detect specific DNA, northern for detecting RNA, and western blotting is for detecting specific protein molecules. So the southern blotting method was developed by scientist Sir Edwin Southern. He was a professor of biochemistry. So this technique is basically used for transferring DNA molecules onto the membrane and further its detection. There are different steps to it, such as separation by gel electrophoresis, transfer onto the membrane, and hybridization with a specific probe. And further, we have the development of the probe. This method is routinely used in molecular biology for detection of specific DNA sequences in the sample. So the DNA that is detected can be a single gene or it can be a part of a large piece of DNA such as a viral gene. So the first step is you have your DNA fragments and you separate the DNA fragments by agarose gel electrophoresis. You also need to run a ladder in one of the lanes, and then you can have different types of DNA samples in different lanes. For example, if you are comparing different types of bacterial cells, that way you can have different lanes. Or if you are maybe digesting your DNA with different restriction enzymes, so that way you will have different samples and different lanes. You perform the gel electrophoresis that is going to lead to separation of the DNA fragments. So here we have the assembly which is used for blotting or transferring the DNA fragments from the gel onto a membrane. The commonly used membranes includes a nylon or a nitrocellulose membrane. So this is your setup. At the base, you have a glass plate. On top of the glass plate is present a filter paper wick, which is dipped in these beakers or vessels that contain buffer. So on either side, they are dipped in the transfer buffer. Then we have our gel that is kept above the filter paper wick. And on top of that, we have our membrane, either nylon or nitrocellulose, followed by a stack of filter papers and followed by another stack of tissue papers. So these papers are basically for allowing capillary action that is going to draw the buffer in the upward direction, forcing the DNA out of the gel and towards the membrane. On the top also we put one more glass plate, and on top of that we put some kind of weight so that the entire assembly becomes compact. So we have here another diagrammatic representation of the same setup. You have the base and then the filter paper wick that is dipped in the buffer that is going to draw the buffer. Then we have the gel followed by membrane, filter paper stack and tissue paper stack, glass plate and the weight at the top. Okay, this, however, you don't have the gap that you can see here. Everything is kept in a compact manner so that by capillary action, the buffer will move in the upward direction. And because of that, the fragments will move also in the upward direction. And in this way, they will be then transferred on the surface of the membrane. So this method of transfer is basically known as capillary electrophoresis. However, nowadays people use other methods like electroblotting. Since these methods, the standard method or capillary electrophoresis is a very time consuming method, the setup has to be incubated overnight or even for 24 hours. before proceeding to the next step. So instead, nowadays people make use of the electro blotting apparatus for transferring the DNA fragments from the agarose gel onto the membrane. So this is the electro-blotting apparatus, which contains a cassette that you can see over here. There are two sides to the cassette, the black side and the lower side is the white side. And they are attached at one end like by a hinge-like structure. So you can open the cassette from the other end like a notebook. And after opening, you have a thin layer of sponge, followed by your agarose gel, followed by the membrane, and then again a sponge on the other side. And after that, you close the cassette just like closing a notebook, and there is a clip on this end to keep it tight. And then you transfer the whole cassette into this assembly, and then you transfer the whole assembly into the electrophoresis tank. Okay, so here in the tank, you have connections, one towards the DNA, other towards the buffer. So the flow of current takes place in such a way such that DNA is negatively charged. So DNA has to move from the membrane, from the gel onto the membrane. Okay. So electroblotting method takes less time. About 2 hours of electroblotting method or electrotransfer method can be enough for transferring the fragments from the uh, gel onto the membrane. Then coming, so this is just steps that are represented. You have your genomic DNA, that is the starting material. You digest, that is going to give you different fragments to electrophoresis that are going to get separated according to size. And then you do a denaturation step by treatment with an alkali. So this is required so that your double-stranded DNA gets converted into single-stranded DNA, which is required before the transfer to be done. Okay. Now, after that, you set them up in the assembly or you do an electroblotting method. So denaturation will have two benefits. First of all, the transfer will be better onto the membrane. And secondly, it is going to allow binding of the probe that is added in the next step. Okay. So at the end of the process, this is what your membrane looks like. You're going to get the DNA fragments from the gel transferred onto the membrane. And then you have a step which is known as immobilization step. So these DNA fragments should permanently stick onto the surface of the membrane. Okay, so before the next step, that is probing, what you do is you do a treatment with ultraviolet light, and that allows bond formation between the DNA molecules and the membrane molecules, and that is going to allow permanent binding of the DNA fragments onto the surface of the membrane. Then the next step is hybridization. So hybridization basically means forming hybrids with the probe. The probe can be either single-stranded DNA or RNA also can be used. It needs to be labeled. Radio-labeled probes were commonly used for the probing method. So you just mix, you just have a buffer present in which you keep your membrane and you add the probe, and then you just keep it in shaking conditions on a rocker basically, so that the membrane is continuously exposed to the probe. Okay, the process generally takes for about 1 or 2 hours, and after that you have a washing step. So washing step is going to remove any non-specifically bound probe. So probe can sometimes also bind to the, maybe the membrane itself, it is binding. So it is not exactly binding, it is maybe just present on the membrane. So we need to remove these non-specifically bound probes that might be present on the membrane. This is done by washing step with the buffer. Then After washing away the excess probe, the last step is autoradiography, that is development of an X-ray film in order to detect the specific DNA fragment. So what you make use of over here in autoradiography is a lead cassette. So it is also like a notebook, you can say, and the walls of it is made up of lead. So in the cassette, you keep your membrane on one side, and on top of that, you put the X-ray film, and then you close the cassette and let it stay for a couple of days. And after that, when you open it, you will see that the impression of the wherever the radioactivity is present, you will be able to see a dark band that has developed on the X-ray film. So by comparing the film with the original, with the membrane, and then with the original gel, we will be able to find out specific DNA fragments or identify those specific DNA fragments. So here we have a summary of all the different steps that we use in southern blotting. We first need to isolate genomic DNA, restrict with specific restriction endonucleases. You can use different restriction endonucleases for digestion in different lanes, or you might be using the same restriction enzyme but having different types of maybe samples or starting materials in the different lanes. So you perform gel electrophoresis that will lead to formation of the banding pattern. Then you perform an alkali treatment that is going to denature the DNA, convert it into single stranded form. Then you proceed to the blotting step where you keep the membrane and either do a capillary electrophoresis or a capillary blotting or electroblotting method that is going to transfer the fragments onto the surface of the membrane, generating a pattern. And further, they are immobilized by UV treatment. The next step is you incubate the membrane with a radio labeled probe. So the probe is going to specifically bind at the DNA sequence that is complementary. And then after autoradiography, you're going to develop the band where the probe has attached, and then you can compare it with the original membrane as well as you can compare it with the original gel, and you will be able to identify the DNA fragment, which you can cut out from the original gel. You can use it for, you can purify the DNA from the cut DNA fragment, and then you can use it later for sequencing to identify what sequence it is, or you can do a cloning or whatever experiment you want to proceed with it. Applications, southern blotting are used for various applications such as gene discovery, mapping, evolution and developmental studies, diagnostics, and forensics. They can be used for identification of transferred gene in transgenic individuals. Southern blotting is used to detect specific DNA fragments, and they can also be used for analysis of genetic pattern that appears in a person's DNA. Then coming to the next technique, that is northern blotting. Now, the other two techniques, the northern and the western blotting, they are basically used since they rhyme. The names are basically used since they rhyme with the first name, that is southern, and they are basically, the other techniques are not in accordance to the name of the scientist. Northern blotting technique was developed by James Alvin in 1979. So it is for detecting specific RNA fragments. So you can do developmental studies, expression studies to identify expression of specific genes and so on. One of the issues with a detection of RNA molecule is that they are very much susceptible to degradation compared to DNA, so they are difficult molecules to work with. Coming to the steps of the method are quite identical to that of the southern blotting. The first step is you do a agarose gel electrophoresis. After migration, you are going to have separation of your bands, And then the next step is blotting, where you are going to blot or transfer the fragments onto a nylon or a nitrocellulose membrane. So you can do a electroblotting or a capillary blotting method here. Whatever is available or suitable can be done. And this is going to generate a pattern of the RNA fragments onto the membrane. Next step is hybridization with a probe. So here also you can make use of single-stranded DNA as a probe can be used, and it needs to be again labeled in some way, maybe radio labeled or chemically labeled or fluorescently labeled, whatever can be used. Okay. After incubation with the probe for about 2 hours, in that is incubated in shaking conditions, the next step is washing to remove any excess or unbound probe again, and then only the specific RNA fragments will be bound by the probe. The next step is again development. By you can use different methods for development. Autoradiography can be commonly used, and it is going to, by autoradiography you are going to get the bands which you can again compare with the original RNA in the agrogel electrophoresis, and you will be able to identify specific gene expression. So what is it used for? It is used for gene expression studies. It can be used for detection of the messenger RNA transcript size. It can be used for studying RNA degradation, splicing, and its half-life. It is also used to confirm generation of transgenic or knockout mice or other animals. Coming to the disadvantages of Nathan blotting, this method is relatively less sensitive compared to other methods that can be used for RNA detection, such as the nuclease protection assay or the RT-PCR. Then also, RNA samples are very difficult to handle. They are quite easily degraded by RNases, so it is a very tedious method because of that. Now coming to the third method, that is Western blotting. This method is used for detecting specific protein sequences, and the probe that we make use of in Western blotting is rather different compared to what we use in southern or northern blotting. In southern or northern blotting, we make use of single-stranded DNA or RNA molecule as a probe, but here we make use of antibody molecules that are specifically raised against a particular protein, and they are going to allow detection of a protein of interest from the mixture of proteins. So here in this method, we also need to separate our protein fragments. The method that is used for separation of proteins is known as SDS page method, which is prerequisite for the Western blotting technique. So this is another difference. In the other two methods, we make use of agarose gel electrophoresis. Here we use SDS polyacryl amide gel electrophoresis. So here, first we do the electrophoresis for separation of the protein segments, and then again we need to do electroblotting method for transferring the separated protein bands onto the membrane, such as a nitrocellulose membrane. Then the next step is incubation with a generic protein such as milk powder can be used or a BSA can be used. So these molecules, they basically mask the surface of the membrane. So the antibodies have more tendency to bind at random locations. So if we do a pre-treatment with milk protein or BSA solution, they basically mask the entire remaining membrane, and this decreases the non-specific binding of the antibody molecules. After making use of the generic protein, we need to again do a washing step to remove excess of the milk proteins that are attached to the membrane, and then we add our antibody. So the first antibody we add is known as the primary antibody, which can also be called as antibody 1. And here, by specific interaction with the protein molecules of interest, the antibodies can bind to the protein. Then coming to the next step is washing again for removal of any non-specifically bound primary antibody. Okay, after that we add our secondary antibody, which is also known as antibody 2, or it can be called as anti-antibody, and it is complementary, or you can say it recognizes the FC domain of the primary antibody. So the secondary antibody that we make use of needs to be labeled. It can be either radio labeled, or or chemically labeled, or it can be enzymatically labeled, fluorescently labeled. Anything can be used. So here we can see an example where the secondary antibody is linked with a reporter enzyme, and then we add a substrate for the enzyme that leads to formation of a colored end product because of the enzymatic reaction. So wherever the target protein is present, it will develop, or you will be able to visualize it as a colored product because of the activity of the reporter enzyme that is linked to the secondary antibody that is bound to the primary antibody, which is bound to the target protein. So this is the image after development of a Western blot. You can see these various uh, bands of the proteins that are developed, and these are the ladder or marker protein bands. So coming to the applications, there are various applications such as in various disease diagnostics. They can be used for detection of specific molecules such as in HIV test or in any other form of disease testing where a specific protein molecule is involved. They can be detected by Western blotting method. So that's it about different blotting techniques. Kindly like, share, and subscribe if you like the content.